And that was a terrific introduction, Cameron. Let me see if I can get the sharing going correctly. Are you seeing what you would expect to see now? You're muted, Cameron.
No. Yes.
Yeah. Okay. That's the view you want. Okay. Terrific.
Yeah.
All right. So I wanted to start out by just acknowledging that originally this is a talk that I was going to give a year ago in Cape Cod, I guess. But of course, as we know, let's see if we COVID came along and canceled that meeting. And so it's now a year later and we're finally getting around to this, although virtually. So it really is a big honor to get this award. I look at the list of the previous recipients, and it's really an impressive and distinguished list. And there's no society that's been as central or important to the development of my career as IBANG's. I have not gotten to all of the meetings over the years, but I've gotten to a lot of them. And it really is the one where I feel like I'm at home and sort of with my family. And so it's very meaningful to be acknowledged by a society like that. As some of you may realize, I was a president of IBANG's many years ago. I can't remember exactly, but maybe 8 years ago. And as I commented during the business meeting, it always was my hope that there would be a stronger relationship between the journal, which is really extremely successful, and the society. And so in thinking about how IBANG's has sort of touched and helped to shape my career, I realized that the journal was really a big part of that. So I was an associate editor for 9 years. I miss having that role, which I stepped away from a couple of years ago. And over the years, have published 20 papers in the journal. So at the rate of about one per year since the journal came into existence. And so today I'm going to review my career very much through the lens of those 20 papers. So sort of a retrospective look. Here are covers. I don't think these actually one-to-one correspond to the covers that were on the particular issues where I published. But it's going to be 20 G2B papers in an hour, and using that as a way of sort of explaining the arc of my career. So the very first one is when I was at Columbia as a postdoc with Conrad Gillum. And Conrad was a friend of Lorraine Flaherty, who was very active in starting the society. And she had recruited him to submit a paper to Genes, Brain, and Behavior, which was then a brand new, I think it was originally supposed to be in the inaugural issue. And for a number of reasons that many of you can relate to, the paper was delayed and ultimately ended up in the second rather than the first issue. But this was bioinformatic work on autism. Conrad's lab was very central at the time to the efforts to use linkage, to use family-based approaches to try to identify chromosomal regions that were associated with autism. And that was a tough thing to do. We now know that the autism is due to many, many very small genetic effects, as well as many de novo effects that wouldn't tend to recur across families. And so that allelic heterogeneity made it difficult for linkage to succeed. But in this paper, we were looking at some recently published linkage results, which identified with at least suggestive significance a number of chromosomal regions that might have been associated with autism. And then we used what was at the time a very new approach, and that was to use bioinformatics to try to relate the genes that were implicated, and I think those are being shown in brown and red, to try to relate the genes that were being implicated to one another and then also to additional genes. And this was all based on what was a new approach at the time being developed at Columbia, which was natural language processing of all of the published literature. So it was all abstracts that were on PubMed at the time, just the abstracts, not the full papers. But then NLP had been used to try to extract relationships between genes and processes and neurotransmitters. And so you see here, we particularly were interested in the serotonin transporter, which at the time was a big story in autism. And so that was somewhat formative in my thinking about network analysis. And just to kind of zoom to the present, now as I have a large project that works on rat genetics, we've enlisted Trey Ideker, who's also here at UCSD, who is working on very evolved, but essentially thematically similar approaches to network analysis. with the exact same idea, that we want to take genes that have been discovered both in humans and in model organisms, particularly rats. We want to throw the things that we're reasonably sure of onto the network, see how they relate to one another, and then see also if we can use that to identify overlapping parts of the network where we see signal from both humans and a model organism, as I said, mostly rats. And we're having some success with that because in the intervening 20 years, we actually know with confidence a lot more about a lot of these phenotypes than we knew back then. Here, this approach was hindered by a lot of things, but one of them was that we were going in there without much confidence in the genes that had been implicated in autism. This first publication really was formative to my thinking, and is a theme that's continued in my work. The next publication comes a few years later in 2006. This was work that actually I had been involved with several years earlier when I was still in the lab of Tamara Phillips at OHSU, the first postdoc that I did, as you heard in Cameron's introduction. I didn't have any good pictures of Tamara, unfortunately, from that era. And the one picture I do have is when she came along with the whole lab to the going away party that I threw for myself because it was clear no one else was going to throw it. When I was leaving Portland in 2002. And so as you can see, Tamara didn't want to be photographed, but what happens when you block a photo is this is the only photo someone has of you, and then it comes back to haunt you many years later. And so this was work looking at the relationship between ethanol and allopregnanolone. Now, allopregnanolone at the time was a pretty obscure endogenously produced ligand for GABA A receptors. Just recently, many of you are aware that actually the FDA has approved the use of exactly this, allopregnanolone, given a different name, of course, for the treatment of postpartum depression, and it's being evaluated for the treatment of treatment-resistant depression also. But at the time, this was a relatively obscure compound, and there was some thought that maybe allopregnanolone might be functionally related to the effects of alcohol on the CNS and on GABA A receptors. And so Tamara's lab, with my help, but some of this done independently by Tamara, had looked at locomotor response to both ethanol and allopregnanolone in recombinant inbred strains previously in an F2 population, which is shown here, and done QTL mapping. And then, as you'll see on the next slide, also in congenic strains. So congenic strains that tried to capture this region on chromosome 2 that was implicated for both ethanol and allopregnanolone, and actually showed that the congenic, so this is a D2 with a little introgress region, actually not so little, but an introgress region on chromosome 2 from B6. And you see that makes the animal less D2-like and more B6-like, and reciprocally a B6 animal and then a B6 animal with an analogous sized region on chromosome 2 from D2, and it makes the animal more D2-like, as if that was an interval that was carrying a critical factor that governed the response to both ethanol above and allopregnanolone below. And so putting all that together, and this was actually the kind of work that I had seen Tamara talk about when I was still a graduate student, and that made me want to go to work in her lab, putting together evidence that came from recombinant inbred strains, from F2s, and from these congenics, we were able to get very strong evidence. You can see extremely significant p-values suggesting that this region of chromosome 2 was involved in influencing the response pleiotropically, sort of influencing the response to both ethanol and allopregnanolone. So around this time, I moved from Columbia to the University of Chicago. And here's a picture of the University of Chicago, not on a snowy day. And we were over in this building here. That's where I was for 10 years. And actually, my house was just sort of off the screen over here. So it's a very, very beautiful campus and wonderful place to be. The first thing that came out of my lab in G2B after moving to Chicago was actually work that had been started in Conrad's lab at Columbia, and with a graduate student, Christine Ponder, who had started out as Conrad's graduate student. And I think it's fair to say that by the end of her thesis, she had become my graduate student. In particular, I remember I was paying her salary. So that's one way you know that somebody's in your lab. So here's a picture from back in the day of Christine Ponder. And Christine and I embarked on a pretty ambitious project to make short-term selected lines for fear conditioning. And this was part of a program project grant that had been going on at Columbia that involved, luminaries like Eric Kandel and Abby Fire and Myrna Weissman, as well as Conrad, to try to look at fear learning and to try to understand sort of genetics and neural circuits associated with fear conditioning. And so as part of that, Conrad had encouraged me to borrow techniques that came from Tamara's lab and from OHSU to make short-term selected lines. So essentially, animals were trained to associate a context and a tone with a shock, and then they were tested for the response to the context, and then subsequently on a third day to the tone. And we selectively bred the animals, breeding the animals that had high fear learning with one another and the ones with low fear learning with one another for 4 generations. And you see that after 4 generations, the high ones were very good at learning this fearful association, and the low ones were very bad, so that we had generated about a 3-fold difference in the capacity for fear learning in these selected lines in just 4 generations of selection. And so this really built on training that I had from Tamara and in Oregon. And we did some cool things with these animals, with that, taking animals from the last generation of selection that were treatment naive, but we knew were genetically predisposed to have high or low responding. We looked to see if varying the number of shocks would alter their response to fear learning, but it didn't. The high animals were always better than the low animals. No matter how many times you shocked them, the low animals would never become equivalent to the high animals. And similarly, if we changed the intensity of the shock, so whereas this was the selection intensity, as we went to higher intensities, we kind of reached a plateau in both the high and the low selected animals, but at no point did we see that a higher intensity would bring them together again. So they really were quite different in ways that couldn't be overcome by changing some of those parameters. We also then looked at fear potentiated startle. So this is a form of fear learning in which, unlike fear conditioning, where the animal has to freeze to show you that they're afraid, these animals have to jump to show you that you're afraid. And we showed that, again, the high selected animals had a really good capacity for fear learning in this very different paradigm, whereas the low conditioned animals did not. These are some control groups that we needed to have to make sure there wasn't a bias in just the responding to the tone. And then finally, Christine showed that these animals, although they had been trained for learned fear, they actually were also very different for unlearned anxiety-like behavior. And that was done with both the open field and the elevated plus maze, and using some of the kind of standard measures in those that I think many people in the audience are familiar with. And so finally, we, because I had this sort of enduring interest in QTL mapping, we did use a procedure that had been developed by the now late John Belknap to compare the divergence in allele frequencies on particular chromosomes over the course of selection with the divergence that would be expected sort of under the null, assuming that there was just random genetic drift in a population of a given size. And in fact, we built on this and also developed a second method that modeled the specific pedigree and that modeled recombination through that pedigree. And the two were in strong agreement with one another, identifying several chromosomal regions that were significantly associated with fear learning. So another example of sort of the drive to identify chromosomal regions. Unfortunately, and I didn't say it, but unfortunately, like with the congenic work that I showed you with allopregnanolone and ethanol, where the region that we identified was relatively large, here also what we discovered is that the regions implicated by these selection processes were relatively large. And in fact, a thing that many people in the audience are now aware of, but wasn't as obvious then, the regions that are most strongly under selection will diverge in allele frequencies in the earliest generations, and therefore they won't have opportunities to have meaningful recombinations, that is, recombinations with the alternative allele. And so in fact, the regions that are strongly under selection will be the largest blocks, the largest non-recombinant blocks, so the least mapping resolution that you could obtain. And that's one of the well understood now limitations of short-term selected lines. As a tool for mapping, I think they remain a fantastic tool for kind of understanding the relationships of phenotypes to one another. So now yet another, now it's 2009, and here's a publication featuring IBANG's current president, Cameron Bryant, as the first author. As he said, he joined my lab in 2007. And here we were looking to identify QTLs for psychostimulant and opioid activity, I think leading with psychostimulant and following up with opioid, which was already an interest of Cameron's. Here's a picture of Cameron in 2009, so the same year that this paper came out. This was actually after the CTC meeting. So in this study, we used chromosome substitution strains. So these are strains that, again, many of you are familiar with, where a single chromosome has been bred in from a contrasting strain. So the animals are all AJ, but with a single chromosome that's come from B6. And so we tested each of these individual strains to first identify which chromosomes were harboring QTLs, that is, which chromosome substitution strains were different from B6. And I think I said it wrong a second ago. They're all B6 with one AJ chromosome. So which ones were different? So you can see, for instance, here and here are chromosomes that are very different from B6. And so that one chromosome changes the phenotype of the animals. And so we looked at that on 2 days of pre-treatment and then a third day of treatment with methamphetamine. We then homed in on chromosome substitution strain 11. Chromosome 11 is a particularly strong effect. And we were also encouraged that it didn't change the innate behavior of the animal in the absence of drug. It only changed the response to methamphetamine. And then Cameron, as I said, went on to look also at the response to fentanyl, which was also different in these chromosome 17 or chromosome 11 substitution strains. And we then made an F2 cross between pure B6 and this chromosome 11 substitution strain to get some recombination on chromosome 11. And we're able to begin to define the critical region for that QTL. But notably, that critical region was about 20 centimorgans, or probably approximately 40 megabases. So still very large and not particularly amenable to gene identification. We'll come back to that in a few slides. The next paper that I'm going to talk about really represents the beginning of an important direction for my lab. So as I was setting up my lab in Chicago, I was focused on QTL mapping. I wanted to identify genes that were associated with behaviors, as Cameron said. But we were using two approaches. One was the sort of traditional approach, which was to use leads that might come from recombinant inbreds or F2 crosses, or to use chromosome substitution strains to identify really big chromosomal regions, and then to try to narrow them down with F2 crosses and congenic strains. But as I set up the lab, I was also really excited about an alternative approach, which was to use populations that had been bred for a long time and therefore had accumulated a lot of recombinations over time. And those populations should, in principle, allow us to get much more precise QTL mapping, because for a marker to be associated with the causal allele, of course, we don't know where the causal allele is. In an F2, the marker could be very far from the causal allele and still be associated. But after all these recombinations, it'll only be the very close markers that are still associated with the causal allele, and thus that show a good p-value. And so just as I was setting up my lab, I was really fortunate to get from Jim Chevrod advanced intercross line, a cross between the LGJ and the SMJ inbred strains. And he had already intercrossed them for 33 generations. For a long time, he had been mixing these strains. And so the chromosomal regions were already broken down into little haplotypes. And Caitlin Samoa, who's pictured here, and Jian Chen developing kind of the software tools. But Caitlin Samoa was an undergraduate. She actually had been a high school student in Jim's lab. And then when she was going to college at University of Chicago, maybe he or she, I can't remember, maybe both of them, contacted me and said that she would like to continue working with these large by small advanced intercross lines. So she actually joined my lab the first week of her freshman year and worked in the lab all through her undergraduate, all 4 years, and collected the data that I'm about to show you, analyzed the data I'm about to show you, and then wrote the paper. She also took the class that I offered that at the time had some undergraduates, so it was mostly a graduate course. And she got the highest score on the final exam in that class, even though she had food poisoning and looked like a ghost when she came to take the exam, but still did better than any of the graduate students or the other undergraduates. So really very talented. She went on to work in Mark Daly's lab and has gone on to have a very productive career actually in autism genetics. I believe now is going to start her own lab at the University of Utah in any time now. I believe that's been finalized. Here's also a picture of the lab at about that time, and you see Caitlin, you see Cameron, you see some other people who I'll mention. Here's Amy Hart. I happened to notice that was her 31st birthday today. This is some time ago. And here's Caitlin actually when she came back from graduate school, and here's a beard. I think you'll see a few photos of me bearded, which I thought would amuse the audience. So here's a slide about advanced intercross lines. I just described them to you, but what's notable about this and the next slide I'm going to show you is these are slides that I used in my talk in 2009 at the IBANGS meeting in Dresden, when I was talking already about this work with advanced intercross lines. So this is literally the slide that I showed in Dresden, just describing the advantages of intercrossing them to the F2 and then continuing to cross to break things down into smaller pieces. And this is the slide that I showed in 2009 to say that we had to develop our own software to deal with the fact that here's the pedigree of these animals from their inception as F2s to the F34s that we were working in. an incredibly complicated pedigree relationship. And it turns out that you need to account for that complicated relationship in order to do a QTL analysis in that population. And so Jian Chen, who I showed a few slides ago, developed the software that he wrote himself to do that analysis. The animals that Caitlin had tested were actually, unfortunately, mostly F2s because of the scheduling constraints of her being a full-time student. But we did know that the smalls and the larges were very different from each other. So these are the pure inbreds that gave rise to that population. And we're looking at pre-pulse inhibition, which is the behavioral trait I won't discuss that this paper was about. And you see that the smalls LG inbred strain are very different from the large inbred strain. And so not surprisingly then, when we did the QTL mapping, and this was with a combination of F2 and F34 animals, we were able to identify some strong signals, but those signals were still relatively broad because predominantly it was F2 animals that were used in that analysis. Okay, so Cameron also talked about work that I've started to do in humans, and that really was made possible by my collaboration with Harriet DeWitt. Now, Harriet DeWitt, I originally met when I was an undergraduate at University of Chicago, and I took a class called psychopharmacology from Harriet and Bill Wolverton. And it was a fascinating class, really sent me off in a particular direction. And I was then excited to come back to University of Chicago many, many years later and to have the opportunity to collaborate with Harriet DeWitt and to sort of be the geneticist for her ongoing laboratory studies. And so she had studies, really fascinating studies, where healthy human subjects would come into the lab and be given various drugs. She's recently published studies with MDMA, but at this time she was doing amphetamine. And then the people would be asked for hours and hours after getting the drug how they were feeling. And in fact, these were double-blind placebo-controlled studies. So they were within subjects. Each subject would come in several times, sometimes getting placebo, sometimes getting various doses of amphetamine. At the time, there was a widespread naivete about the effect sizes that were realistic to expect in human genetic populations. And so with that in mind, Harriet's lab wanted to look at some of the prominent candidate genes of the day, including OPRM1, the opiate receptor. And so we helped her with the genetics of that. Oh, and here's the first author. This is Andrea Dlugas, who's the first author on this paper and was a postdoc with Harriet DeWitt. And she's actually holding Ariella Palmer, my daughter, who was obviously very young at the time. Now she's 12 years old, and I believe might be taller than Andrea is now. She's grown dramatically. And here I am with Harriet DeWitt, and no beard, but at least some sideburns to keep people entertained and engaged. So here's the OPRM1 receptor. We looked at just a few SNPs that were meant to tag the majority of the coding region and the introns of OPRM1. Here, notably, is the A118G polymorphism that we heard about in earlier talks. And you'll see that it's in perfect LD with this SNP. What we reported was an association of this SNP, but you see it could just as well have been annotated as 111799971. We saw an association between that polymorphism and the response to the 10 but not 20 milligram dose of amphetamine. Now, looking back at this, I feel that I'm not very convinced by it anymore, because we now know that the effect sizes of alleles are small enough that in a population, I haven't said yet, but this was a population of about 150 individuals, you wouldn't really expect to see such a large effect. And so this is going to be mostly winner's curse, even if there's something real here. And when something's mostly winner's curse, that means there's a higher than the p-value would indicate risk that it's actually a false positive. Be that as it may, it is interesting to note that now the only genome-wide significant finding for opiate use disorder in much larger, very well-powered human genetic studies is in fact the OPRM1 gene, and in particular is that 1799971, that A118G polymorphism that is essentially being tagged as in perfect LD with the SNP we reported here. So there is some evidence that that particular SNP really is important for some phenotypes, but keep in mind, this is a much larger sample. It's a very small effect size in terms of the beta for the effect size. And this is for opiate use disorder. Nobody obviously has done a large study of the subjective response to amphetamine and looked at OPRM1, and that's because looking at subjective response is so difficult to do at scale. I think those studies will probably never be done. And there's another paper that came out a few years later, so slightly out of the chronological order of this talk, but this was also from Harriet Zwitt's lab. This was Megan Wardle, who's now back in Chicago at University of Illinois. Chicago as a faculty member, at the time was a postdoc in the lab. Here's the birthday girl, Amy Hart again, who was very involved in these studies. Here, actually, we were following up on reports that COMT polymorphisms influence executive functioning. And what we reported was that we didn't see that. And the studies that had reported that association were in smaller samples. We had a much larger sample. We had repeated measures, and we still didn't see any evidence of that COMT. So this was a part of a trend where initially there were a lot of candidate gene kinds of studies, very underpowered, that showed exciting positive results. And then there were a lot of non-replications published, which struck a cautionary note. And in fact, Harriet and I, with Amy, this time as first author, published a paper not in G2B, sorry for the intrusion, but in Neuropsychopharm, where we said that after collecting a few hundred more subjects in Harriet's paradigm, not just a few, but none of the initially published results could be replicated, even though we had a larger sample size and should have had more power to replicate them. So this was our very cautionary message about this candidate gene approach in 2013. So now going back in time slightly to 2011, but thematically related, I want to talk about a few papers. This one headed by Greta Sokolov, who was sort of the lab manager for me in Chicago for several years, is now at the University of Iowa. And actually, I've been trying to court Greta to maybe come out to San Diego. Who knows if that'll be successful or not. But this was her following up on some data that Jackie Lim had collected in an F2 cross between C57 and DVA, looking at anxiety and fear-related measures. So here's Greta and Cameron and me, again, some sideburns to keep everybody entertained in front of a fake backdrop of the London Bridge. And then here, contemporaneous with this 2011 publication, are people that went to the IBANGS meeting in Rome. So here's Cameron, there's Clarissa, and here's some other people from my lab in Rome at IBANGS. I actually was unfortunately not able to go to that meeting. In this paper, we started to think about the temporal relationship for QTLs. And so what we're doing here is we're looking at the physical position on one axis, that is marker positions on a particular chromosome. We're looking at the LOD score, the strength of association, so higher is more strongly associated. And then we're looking temporally throughout this fear conditioning paradigm, basically exactly the same paradigm that I mentioned earlier with Christine Ponder's paper. And what you see is that there's a particular chromosomal region which is important late in the paradigm during the tone sessions, but not important early in the paradigm before those tones that have been paired with shock have been presented. And so this visualization of the sort of temporal nature of a QTL and how it would affect behavior at certain times during the test, but not others, was our big advance in this paper, I think. We also did some sophisticated work that I think I'll glaze over in the interest of time, but where we built out a model. And so we built out a model that had multiple QTL effects modeled in it. And that actually allowed us to identify putative interactions between loci, and it also allowed us to identify regions or chromosomal regions that weren't significant initially, but were significant when we put in enough of the other QTLs into the model. So as we improved the model, we were able to detect more significant effects. Another related paper that came out in 2012, Clarissa Parker is the lead author here. This is looking at methamphetamine sensitivity in advanced intercross lines. This was actually a population that we housed only briefly, and these were a B6 by D2 advanced intercross lines. And we actually sent some of these off to Rob Williams and Lulu years ago, and they've used them to make recombinant inbred strains that are derived from advanced intercross lines, so have a higher density of recombination than your average recombinant inbred line. Here's Clarissa. This is actually Sam, my second child, with Clarissa and me in our kitchen in Chicago, in my kitchen in Chicago. And then here also Clarissa at that IBANGS meeting in Rome, so slightly out of step, and Clarissa again in Rome. And so in this paper, again, QTL mapping, this was in, again, an advanced intercross line, and we were able to get relatively high resolution and extremely significant associations. And again, we used this three-dimensional approach to look at the relationship between position on the chromosome, temporally, that is within the experimental paradigm, and then the significance on the y-axis. And so you see that we were able to find really a lot of very significant effects, some of which were very temporally specific, so influenced only a certain part of the behavior. And then another paper, this one from Cameron Bryant in my lab, and this is congenic dissection of the region on chromosome 11 that I mentioned a few papers ago that Cameron and I had found in the chromosome substitution strains. So here's Cameron. I've now shaved off the beard and the sideburns, and I look very excited about that, kind of bizarrely excited. Here's the lab approximately contemporaneously with this. And this paper came out in 2012. Here I am at IBANGS in 2012. This was in Boulder, Colorado, up in the mountains. And you see the beard. So what happened here is one of the reasons that I had already been concerned about congenic lines, but I think one of the things that really solidified for Cameron and I our concern about congenic lines. We made a number of congenics. So these are regions that are entirely B6, but have a little introgressed region from AJ, and they were overlapping. And the idea was, what is the minimally sufficient region to capture the QTL? And we found that line 1 captured the QTL, that is, it was different in its response to methamphetamine. And line 2, I think, also did. But when you look at these and you put them all together, you realize that it doesn't make sense, that there isn't one critical region, that instead it seems that there are multiple critical regions within this region. So what had looked to us at low resolution like it was a single QTL was actually probably a constellation of genetic factors that, as they were broken apart by this congenic dissection, sort of fell apart and stopped being one thing and turned into a number of things. And so that's really a cautionary note for that congenic approach, which does sometimes work. Sometimes the thing that you see really is one cohesive thing, and you can narrow it down, and eventually identify it. But it's equally likely that there are multiple genetic effects that are close enough to each other that they initially look like one thing, but that as you narrow down on them, they actually turn out to be multiple things. And so a similar story here, this time led by Clarissa Parker, looking at fear conditioning. And so now this is a 2013 paper. So here's 2013 in Belgium, and several people, Iris and Stephanie and John, some of you will recognize. And here's the part of the lab in 2013 at a different meeting. So here again, we were using F2 signal to lead us to a particular region that we tried to dissect with congenic lines. But when we started to dissect it, we found that both line 2 and line 1 captured part of the QTL. So line 1 captured more of it, but line 2 captured some of it, but significantly less. And so in fact, this region wasn't a single factor, it was at least 2 factors that we were able to begin to dissect. And the trouble with that is that as you start to break apart this initially big signal into smaller pieces, the signals get smaller and smaller and therefore harder to work with. And the sample sizes required become larger, and that's not what anyone would want. So really, in those last few papers, underlining the limitations of that approach. That said, though, Cameron did have other stories that were extremely successful. And in fact, he carried on work that was based on congenics that he had done in my lab and did successfully clone a gene and has now done a lot of work on the biology of that gene. And Cameron, I apologize, I can't remember at this instant the name of that gene, but at any rate. And then one last congenic paper, and this was in collaboration with Melody Winower, who's actually a neurologist at Columbia, who I worked with for the first many years that I was in Chicago. And this paper eventually came out. Oh, here's 2014 IBANGS. This was in Chicago. Here we are out on the lake. Chicago could be a beautiful city as viewed from the lake and in good weather. And here are some other members of the lab in 2014 at ACNP. I won't say very much about this, except that this was a relatively more successful congenic story where we were able to dissect down to a very small critical region and identify a very significant effect. And this was a seizure sensitivity QTL that Melody has continued to work on since then. So I've talked a lot about the advanced intercross lines that I used sort of early on in the time in my lab, but I was also thinking about other ways to do things. And so not reflected by any of these G2B papers was the growing interest I had in using commercially available outbred strains for mapping, reasoning that they would have even more accumulated recombinations than we were likely to get from the advanced intercross lines. And actually, when I gave a talk, I looked back when I gave a talk in 2009 at the IBANGS meeting, I talked about the CD1 mice at the time. We then shifted gears to CFW mice, but that was the 2016 Clarissa Parker publication that Cameron mentioned in my introduction. So that was sort of already going on in the lab, was the attempts to use populations with even more recombinations. The other thing that was going on, though, while I was still in Chicago, is that as we were getting more and more successful at mapping things with these highly recombinant populations, like advanced intercross lines and outbred populations, I wanted to look at more and more complicated phenotypes, behavioral phenotypes. And I started to realize that a lot of the behaviors that I was interested in had been developed in rats, and then with varying degrees of success had been sort of ported over into mice. And what I was trying to do was bring the phenotype to mice. And I realized that the other way to solve that problem would be to bring the genetic tools we had developed in mice to rats. And so in these two papers, this one with Jerry Richards as the lead author. Jerry Richards, by the way, was a postdoc in Lou Seiden's lab at University of Chicago when I was an undergraduate dishwasher in the lab. So somebody who I had known for a long time. He was a rat neuroscientist who had never started using mice. And he had really elegant paradigms. So this is showing locomotor behavior, but I'll also show you delay discounting and some other really sophisticated behaviors that he was very good at doing in rats. And I said to him, Jerry, let's look at some inbred rat strains that are approximately the progenitors for an outbred rat population that I'd like to use, and see if they're very different for some of the behaviors that you are familiar with measuring. And so we saw that they were very different in their locomotor behavior. Here are the different strains that we looked at in the locomotor behavior. And here are some statistical analyses showing that they're different from one another. They were also very different in their delay discounting. So this is the tendency to attribute less value to rewards the longer you have to wait for them. And we saw really high significance here that the heritability of this trait was about 60%, if I recall. Jerry said to me around this time that all of his studies to date had been pharmacological. And he said these genetic effects are much bigger and more robust and more consistent than any of the pharmacological effects he was used to working in. So this turned out to be critical preliminary data, as I'll describe in a second. We also worked with Hao Chen using a similar kind of approach, but here we were working with the outbred population. So with the outbred HS rats, which are the product of mixing 8 inbred strains back in 1984, and they've been maintained as outbred ever since. And he looked at them for nicotine self-administration and found that a lot of variability across these strains, which we now know is partially due to genetics. And this was context-induced reinstatement of drug-seeking behavior, of nicotine-seeking behavior, but there was a whole battery that was done. You see this is figure 7. And so those two papers represented critical preliminary data that there was a lot of phenotypic variability and heritability in heterogeneous stock rats, this outbred rat population. And so data from those two papers were critical for the initial P50 grant that we wrote, that we submitted in 2014, and that, as Cameron mentioned, has been now renewed out to 2024. And so that was critical preliminary data that allowed us to move in a new direction, which was no longer working in mice, which had been the basis of my lab for many years, but instead transitioning into rats. And although her name is not on this, Clarissa was really important to some of that work and was the lead author on another paper not published in G2B, kind of making the argument that genetic tools developed in mice could be equally well applied in rats. And we could then take advantage of the more sophisticated behavior of rats. And then obviously here's Cameron. This is at the IBANGS meeting that I think was in Uppsala in 2015. And here's a larger contingent of my lab in 2015. 2015 was also the year that I said goodbye to the University of Chicago. And so here's a sign that we left on one of the boards there. We packed up all of our stuff and we moved to San Diego, and where we've been ever since. Here's a lab meeting shortly after we arrived in San Diego on the beach, not far from my house. And I'll note my children here growing. And a few more papers left to cover. This is one by Laura Sittig, the first paper that came out in G2B after that move to San Diego. And this actually took advantage of an F1 population that was the basis for a paper that got a lot of press. It was a paper in Neuron showing that when we took mutant alleles and we crossed them onto each of 30 different F1 backgrounds, we saw really different effects of those mutant alleles. And this was done with TCF7L2, which was a type 1 diabetes gene, and also with CACNA1C, which is a gene implicated in a variety of psychiatric disorders, principally schizophrenia and bipolar disorder. And we saw in that paper, which I'm not going to talk about, but we saw such differences that in some backgrounds, the genes would increase relevant phenotypes, and in other backgrounds, the same mutation would decrease the phenotype because the background was so important. But we also wanted to use that population. So here's Laura actually at the filming of a sort of amusing video that we did describing that paper. Here's Laura again with other members of the lab. But we wanted to also use that population, just the sort of clean F1s, the F1s that didn't have the mutant allele, as a mapping population. And so this was a paper where we used them and looked at the phenotypic diversity. We had genotypes of these animals, which we knew a priori because they were inbred strains or F1s from inbred strains. So we already know their genotypes. And we were able to identify some significant associations there with those traits. Here's a funny paper. This shows up in 2016. I don't know if Cameron thinks this is so funny. Cameron's the second author on this paper. This is a paper that originally took form in about 2009. And here's Alyssa Chesler with me at the Winter Brain meeting when we were already hatching the plan for this paper that didn't come out for another 7 years. But Clarissa, working with Will, had identified QTLs and recombinant inbred strains on chromosomes 5 and 15. The chromosome 15 was coincident with the gene casein kinase 1 epsilon, which Cameron and I had done a lot of work on, though I haven't described it today. And Cameron and I had this drug, which was an antagonist of casein kinase 1 epsilon. And so Cameron did the von Frey test, spent a lot of hours down in the lab poking those little fibers into animals, was able to show that this casein kinase 1 inhibitor altered the responses on the von Frey test, substantiating the idea that casein kinase might be the causative gene at that allele. But it's just sort of a story about patience because it took from inception to publication at least 7 years. I think it might have even been longer, to tell the truth. And now beginning to wrap up, but here's a paper where Paul Meyer is the lead author. Paul was a graduate student in Tamer's lab when I was a postdoc, and we actually shared an office for a little while and had a lot of fun there. When I started to put together that rat P50 center that I mentioned, Paul was by then at University of Buffalo, where Jerry Richards was, and Paul became a part of that, originally working as a co-investigator on Jerry's project, and now has his own project in the renewed version of that. And this is a paper that we did with Paul's lab about this gene CDH13, and it's the gene that encodes the peptide ligand for that receptor. And we had data from both mice and rats implicating that in Pavlovian drug conditioning. And I think in the interest of time, maybe I'll skip over that because I want to be sure to finish on time. Here's another one. This is from Leah Solberg-Wood's lab, and I'm a sort of a guest co-author. Leah is the one that actually maintains that heterogeneous stock population, that outbred rat population that I keep referring to. This is Katie Hall, who's a phenomenal technician in Leah's lab, who really single-handedly kept the rats shipping for years and years as that P50 center was getting going. And this was sort of her project. By the way, this is contemporaneous with the IBANGS meeting that was in Rochester. I don't know why, but this is the only photo that I seem to have taken while I was at that meeting, was of one of these old, to me, fallout shelter signs that was in Rochester. But at any rate, Leah, working with Katie, had found that animals that had a sort of a depressed-like phenotype, that is, that had really high mobility in the forced swim test, were sensitive to fluoxetine, whereas these genetically heterogeneous animals that had less depressed phenotype weren't sensitive. And so this was an interesting observation that maybe there was some sort of a sensitivity to fluoxetine that could be studied in this population. That's the thing that I think Leah's interested in continuing to work on. Almost done, I promise. Here is a review paper that Sandra led talking about human personality genetics. Here's Sandra actually when she presented other work at IBANGS in 2017, when it was in her home country in Spain, in Madrid. And here's the lab celebrating when she was promoted from postdoc to assistant professor. She's now an assistant professor here at UCSD. And basically, this review highlighted the progress from no genome-wide significant findings for personality to enormous numbers, or even by then, 2017, enormous numbers of genome-wide significant findings for personality traits. And that's only grown in the intervening few years. And it's really the theme in human genetics, which is that whereas people initially hoped that small populations, maybe even 100 or a few hundred individuals, could be used to study genetic basis of behavior, instead it's taken tens of thousands, hundreds of thousands, at this point into the millions in some instances, to get at to get the power to identify the alleles that influence human behavior and personality. But once you have that power, you can begin to identify, initially a few, and then tens, and then hundreds of alleles of very small effect that influence those traits. And so this sort of charts the progress of that, just specifically for human personality traits. And then finally, the most recent publication in G2B, this has just gone online in the last few days, and you'll see a Palmer as the lead author, but that's not me, that's Rohan Palmer as the lead author. And this is describing a meeting that was held in Oak Ridge a year or two ago, I guess 2 years ago, and describing kind of some of the discussions that were had at that meeting about ongoing efforts to integrate genetic studies of behavior in animals and in humans. And that's become increasingly a theme in my own lab, is how can we bring the arc of the work that I've been doing in human genetics for a number of years, first with Harriet and more recently with 23andMe and UK Biobank and other big outfits. How can we bring that arc together with what we've been doing in animal genetics? And in particular, there are a number of traits where we're trying to study the same thing. So the delay discounting work that I mentioned with Jerry Richards, we're also studying delay discounting in human populations from 23andMe, with the hope that we can actually start to have those two co-inform one another. And one of the ways that we're doing that is with the network kinds of analyses with triadic that I mentioned after the very first paper in this talk. So are there any morals or conclusions or take-home lessons from this story? The big one that I think is really important to say, especially in the context of an award talk, is that nobody does this alone. It's terrific to say Abraham Palmer has had a distinguished career, and maybe that's true and maybe that's not true. But any distinguishing features of my career are made possible by all of the wonderful people who've passed through my lab. Happily, many of them have remained involved in this society. Cameron and Clarissa are prominent examples of that. Another one is that one big QTL is often made up of a bunch of little QTLs, and that's why there's really been an increasing emphasis in my lab on using these highly recombinant populations, so that when we do identify an implicated region, it's a very small region. It's less likely to contain multiple causative alleles and more likely to really be one causative allele that we can then go about identifying and cloning. And so that is part and parcel with the idea that having greater precision is really critical for QTL mapping, that the kind of selected approaches, the F2 crosses, some of the things that I was talking about 10 and almost 20 years ago are not optimal. They were possible then when other kinds of studies weren't possible, but the optimal study is always the one that emphasizes precision of mapping, even if that happens at the expense of other priorities, including sample size. Rats are the smart choice, so that's the name of the review paper that Clarissa wrote, and I'm sort of joking, but I do think that for a society that has a lot of people that are using other simpler model organisms, it's important to emphasize that the advantage of rats is the really sophisticated operant and other kinds of behaviors that you can do in rats, and the potential to discover loci that are associated with those complicated behaviors is exciting, I think. And then the last thing is just on a personal note, that it's the people that you meet and interact with, and in some cases train, and in some cases are trained by along the way, that really make this fun and exciting. And I think you saw that there were a lot of arcs of people from earlier in my life coming back at later points in my life. And that really is one of the things that when I thought back has made this career really exciting and fun and rewarding. So that is my last slide, Cameron. I don't know what you want me to do, if you want me to unshare my screen, or if you have some questions in the chat.
Yeah, so unshare your screen maybe. You had a button earlier, just a few minutes ago. My screen is totally messed up now. I don't know about everyone else's. It could just be my computer though.
Okay, I'm sorry to hear that.
Yep,
So
I
have to.
unshared.
Okay, very nice talk, Abe. Sorry for the interruption there. Yeah, I have to promote people to panelists too, so I'm going to try to do both things. Abe, congratulations on the award. Thank you for your service to the society and overall leadership in the field of behavioral neurogenetics. This is Vivek Kumar. Your talk nicely incorporated the evolution. There, okay, fine. It's clear now, I think. Oh, maybe not. Anyway, your talk nicely incorporated the evolution of field of quantitative genetics and its utility in dissecting complex behavior. We're in a new phase in which human genetics has finally matured and is giving us candidate genes underlying complex diseases. Your lab has also embraced the human GWAS approach. Where do you see the utility of model organisms in discovery now? Is there still room for model organisms to lead in discovery, or are we relegated to functional follow-up of discoveries made in humans? Very good question.
That's a great question. I'd love to talk about that. There was a sense early on when I was doing this that maybe model organisms needed to lead the way. That is, we had to find the genes because the human geneticists were never going to find them. They were fooling around with linkage, as I showed in that autism paper at the very beginning. That's certainly not true anymore. GWAS works really well. There are some traits that you're never going to touch with GWAS because they're just not practical to do at scale. But for things that can be scaled, GWAS works very well. That said, I think a gene that's discovered in animals, even if it doesn't turn out to influence disease or other traits in humans, can be really important because it's nevertheless a way to manipulate a system. It's potentially a drug target. It's potentially a way to get involved in some biology, even if it's not biology that you've discovered in humans. So I think discovery of genes in model organisms totally independent of humans is a very legitimate and important pursuit. And I don't think anybody should feel ashamed or bad about saying that in the face of the success of human GWAS. But I do think that another important pursuit that really wasn't possible back then, but is now, and is the use of animals to model disease alleles that are discovered in humans, and then to do that in controlled environmental conditions on an isogenic background with the ability to get into the biology using all manner of modern techniques like DREADS and optogenetics, and et cetera. So that's another huge opportunity that really wasn't something we could contemplate even 10 years ago because human genetics was still mostly a dud at discovering things.
Okay, I don't see any other questions. I think the rest of the chats are all on the upcoming session. Anybody else have questions for Dr. Palmer? No?
Okay.
Okay.
I very much appreciate the award.
Congratulations, Abe.
Particularly gratifying to get the introduction by a former postdoc.
And it was a privilege to give it. Thank you very much.
Yeah.
All right. So we are missing a panelist, and my computer is totally jacked right now. Like it's all blurry. It has something to do with Zoom. I don't know. It's just kind of like, but I think I can
Carl
manage.
should take over maybe for a minute. Can you?
Well, I can designate him, as chair just in case.
And then if you want to just leave and come back, I think we can take like a 5-minute break. We have one talk less. We'll be all right. So if we want,
rebooting would be helpful for me.
Yeah. So if we want to start, well, everyone else that's not having problems can just stay on the Zoom, but we'll start at like 2:08 or 2:10. I don't care.
Yeah, 2:10 is fine.
Okay.
Is it 3:10 or 2:10?
Well, I was using Central Time.
Right.
Sorry.
3:10.
Yes, 3:10 Eastern. Okay, I'll see you guys soon. Thanks, Ed.
Yeah, thanks.